Hello everyone, I am Soma Banerjee, a scientist from a biotech company named Aptologic Incorporation from Ames, Iowa, USA. Thanks for joining the poster session at the Aptamers 2024 11th International Conference in Oxford. In this presentation, I am going to discuss the potential of DNA aptamers to distinguish dengue and Zika NS1 protein. Dengue viral infection is seen as a global threat that necessitates early diagnosis. The current method of dengue diagnosis is based on viral isolation, RT-PCR, and serology-based detection, which are expensive, time-consuming, and requires trained personnel. Besides, prolonged presence of antibodies in the host blood fails to give an idea about the timing of the infection. For early diagnosis of dengue, the NS1 protein is considered as an ideal antigenic target. However, the NS1-based detection primarily depends on antibodies, which suffers from expensive production process to non-uniform batch synthesis. The ability of aptamers in overcoming the limitations of antibodies motivated us to isolate DNA aptamers against the NS1 protein of dengue virus. Both dengue and Zika viruses are positive stranded RNA viruses, and they both belong to the same family of Flaviviridae and are structurally similar with about 11 kilobase genomic size that encodes 3 structural and 7 non-structural proteins. And the similarity further extends to the NS1 protein structure from both of these viruses. The protein sequence of NS1 from both dengue and Zika is about 56% identical. Interestingly, the electrostatic surface potential maps of these NS1 proteins show several virus-specific features on the loop surface of the protein. Dengue virus serotype 1 and serotype 2 NS1 structures display a positively charged surface in the central region of their loop surface, whereas NS1 from Zika virus exhibits both positively and negatively charged central region of the loop surface and more negative charge towards the two distal ends. Therefore, despite the significant structural similarities between dengue and Zika NS1 proteins, the possibility of having viral-specific NS1 protein aptamers cannot be ignored, and some RNA aptamers have already been reported that bind specifically to dengue NS1 and not Zika NS1. A total of 15 cycles of selects were carried out against hexameric dengue NS1 protein that is secreted in the host blood, with successive rounds of counter selections against Zika NS1 protein to drive the specificity of the isolated aptamers towards dengue NS1. The binding percentage of the oligo pool to dengue NS1 over the select rounds show enrichment of the pool with the potential aptamers. When we looked into the specificity of the protoaptamers 6052 and 6067, which are the full-length oligos from the final enriched pool of the selects, we found that these protoaptamers can specifically recognize dengue NS1 protein at concentrations above 250 nanomolar, whereas below that concentration, they cannot distinguish between dengue and Zika NS1 protein. Therefore, our results further opened up the questions like, does the Zika NS1 protein exist in different oligomeric state or structural conformations at different concentration? Why the selected protoaptamers can bind Zika NS1 at concentrations below 200 nanomolar but not above that? Is it because the binding site of those protoaptamers on Zika NS1 getting masked or disrupted at higher NS1 protein concentration? We are still trying to find the answers. Finally, before I end my talk, I thank NIH for funding this project and Professor Marie Nelson Hamilton for her valuable suggestions and guidance throughout this work. If you have any questions, please reach out to me through my provided email ID and I'll be happy to get back to you. Thank you.